This is Photosynthesis Experiment 4, and we are at steps 2 and 3. Step 1, I removed the geranium plant from sunlight for 24 hours. Step 2, we've placed the film over the plant leaf. I made this film from electrical tape, plastic, and cardboard. And we are attempting to answer the experimental question, how is it possible to leave a permanent image on a plant leaf? So step 3 of this experiment is to leave this plant now in sunlight for the next 2 to 3 hours. Continuing on with project part 4, photosynthesis experiment part 4, the geranium plant sat in the sunlight for several hours with the makeshift film covering the plant leaf. So we're going to continue on by continuing that process that we did in previous lessons. I'm going to remove that plant leaf, and that will be the plant leaf that we're working with. So we have our geranium leaf that we have already built up to with work. We have our rubbing alcohol. I did find 2% iodine, so I picked some up. And the first step that we need to do is give our leaf in a shallow bowl a hot water bath. I just got the water boiling. We need to repeat this process 3 times for a minute each while pushing the geranium leaf down to the bottom so it's a little more weighted down in the fluid. I'm just about the end of our next step. I've been pushing our leaf down and changing the water out in 1-minute intervals with fresh boiling water to get that leaf so it's not floating to the surface. In the meantime, I started our rubbing alcohol hot water bath that's supposed to sit for 5 minutes before we transfer the leaf into there to start removing the color. Okay, the rubbing alcohol has now sat tight for 5 minutes in its bath, so I'm going to change out that water with fresh boiling water. And I'm going to add our leaf using the tweezers into the rubbing alcohol and then start our first 15-minute process of swirling and trying to remove the green from the leaf. During the 15-minute time, I will replace the boiling water at least 1 time, and then we'll repeat the process a 2nd time. Continuing on with photosynthesis experiment number 4, we are just finishing up step 8, where we're in our first 15-minute interval of removing the green color from the leaf. You can see that water is turning, or the rubbing alcohol has turned pretty green. So we actually have to replace the rubbing alcohol with fresh rubbing alcohol, and then we're going to replace the hot water, and we're going to do this 15-minute interval again to try to get all of the green out. Okay, we're on step 9. We're repeating this 15-minute interval. I've got fresh rubbing alcohol. I've got fresh boiling water in our cup. Repeating that next 15-minute interval. Well, I'll swirl it, and I'll also change out the water at least 1 time during this 15 minutes. This was the rubbing alcohol from the previous 15 minutes, so you can see we got a lot of that green out. So we'll see how much we can get out in this next batch. We are at the end of step 9 in photosynthesis experiment part 4, where this leaf has just gotten its second 15-minute bath. So we're going to pour out our hot water and then pour out our rubbing alcohol. Quite green. You can see all that chlorophyll came out. And then we're going to give our leaf a hot water bath. said that we should have gotten all the color out. We got a lot of green out, but we definitely did not get all of the green out. But let's go ahead and add the iodine and let it sit for 5 minutes, especially now that I have 2% iodine. I'm actually following the rules better because it said make sure it was 2% and I was using 10%. So I'm going to put a couple of drops of iodine around the leaf. And then I'm going to let that sit for 5 minutes. Okay, we're finishing up. We just finished step 11 where we left the leaf in an iodine. We covered it and then we left it on a plastic tray for 5 minutes. Now I am on step 12 where they ask us to rinse the leaf off and then examine the results. Okay. So, there it is. We can see that there are two wide open areas where it looks like the stickers were placed. So if you compare the stickers, you can see indeed where they were placed on the plant leaf. So I imagine that if we had tried this process for a longer period of time, if I had left the plant in the dark for longer and then perhaps gotten more sunlight on these, then that image would have been even more identifiable on that leaf plant. But we can see that the area that was covered by the stickers was not able to perform photosynthesis and make the starch needed. So once it had used up its starch reserves after being deprived of sunlight for a while, then it started to stop cellular respiration. We can see that because there's no starch in this area that we continued to keep covered. So there was no starch being made, which means there was no photosynthesis occurring in those cells that were beneath the stickers on our film print. Okay, so taking it back, how is it possible to leave a permanent image on a plant leaf? If I remove a plant from sunlight so that it cannot perform photosynthesis, it will use its starch reserves. Then I place it back in the sunlight with some kind of image covering it. The part that is covered will not be able to perform photosynthesis because it's not getting any sunlight. So the cells will not be performing cellular respiration and using that starch to create energy. They're dying. We can see that because there's no stain in those areas where the stickers were covered. Okay?